connected to at least one or two of these, and in many cases, all of them actually. So we're really excited to kind of see these new speakers, welcome them to our community, and then think about how we collectively can move in these new research directions. Our first speaker today is Elizabeth Harvey. She joined UNH in August 2019. Her research focuses on elucidating the mechanisms that mediate associations between marine microbes, with a focus on understanding interactions that result in microbial mortality, the death of microbes. Very sad. Research in her lab involves an integrated approach across multiple spatial scales, from molecules to ecosystems, to recognize the mechanisms underlying shifts in phytoplankton population dynamics and primary productivity, and ultimately to better understand ocean biogeochemical cycles in global climate. She received her PhD from the University of Rhode Island, and with that, I will hand it over to Elizabeth. Take it over, Liz.
Thanks, everyone. Thanks, Eric, for the invite, and thank you, everyone, for being here today. So with my 15 minutes of time, I want to give you sort of an overview of especially some research that I am getting started and want to get further down the road in being here at New Hampshire. It's not a complete comprehensive overview of all the projects I'm working on in my lab. I wouldn't have time for that, but it gives you a little taste of some of the things that we can do in the Harvey lab. So I don't think I have to convince this audience very much that plankton are important. Hopefully you all feel that way a little bit. You all enjoy breathing, right? We know phytoplankton in particular provide you with oxygen, and they're the base of the marine food web. I really enjoy this piece of art here. It demonstrates all the different types of plankton that we have in the marine environment. Clearly we have fish and copepods and diatoms and microzooplankton. Not everything is to size in this picture, although giant copepods like this might be really interesting if they existed. I would be remiss if I didn't include in this sort of microbial soup viruses and bacteria as well. They are also really important microbes in the marine environment. And I know many of my maybe physical or chemical oceanography compatriots in the audience here may tend to think about, you know, sort of the plankton in terms of this, just a box of carbon. And I can appreciate that and I can understand that. But in my research, I really like to look at plankton on the individual level. And so a lot of the work that is done in my lab really focuses on what phytoplankton are there, what are they doing, who are they interacting with, and then what or how does that impact ecosystem structure, ecosystem health, biogeochemical cycling, things of that nature. So we're going to run through a very quick math problem here about what modulates phytoplankton biomass in the ocean. So phytoplankton biomass is really based on the amount of initial phytoplankton biomass that you have, multiplied by the exponent of the net growth rate, multiplied by some time period, right? So we can use this to just very quickly calculate sort of a net growth rate or net accumulation rate of phytoplankton over time. And really this net growth rate is based on two factors, factors that impact growth and factors that impact loss. And so we know a lot in the marine environment about what influences growth, right? Light and nutrients, just like you would spray Miracle-Gro on your garden. If you could do that in the ocean, I don't recommend doing that. But if you wanted to, or you could, you would likely stimulate some phytoplankton growth if there was enough light around. What we don't have as great a handle on is the loss rates. So, you know, physical forces that shuttle phytoplankton to various places, also different forms of mortality that can cause phytoplankton loss. And so what I want to put forth today is some work that I did along coastal Georgia that I think is really applicable and what I hope to bring to coastal New Hampshire. And that is using time series data, and specifically phytoplankton time series data to really understand the mechanisms that sort of mediate these population dynamics. So the shifts between enhanced growth or enhanced loss, and how that really impacts phytoplankton dynamics. So I am going to tell you a story most about Georgia. It's probably a lot warmer there, actually, even though everybody's cold today. It's probably warmer there than it is here right now. Just to orient everyone, I'm going to be talking about work that was done at the Skidaway Institute of Oceanography. So we're down here, right sort of on the border with South Carolina and Georgia, and Savannah, Georgia. The Skidaway Institute is out here on Skidaway Island. It's very beautiful. This is what it looks like at sunset sometimes. And I'm going to be talking about the work of a graduate student I had while I was there, and a year-long data set that we compiled as part of his PhD work. So this is my student, Sean, here, very studiously sampling. And what we did over the course of the year is sample for a variety of different parameters. So your very basic temperature, salinity, dissolved oxygen, chlorophyll concentration, but we also added, I guess I'll say, some flavor on top of that. So we also looked at community composition. We measured microzooplankton grazing rates, viral mortality rates. We looked at organic carbon and transparent exopolymer particle concentrations. And then we also took samples for subsequent DNA measurements after all the sampling was complete. And so I'm going to sort of walk through some of our findings, and we can think about the power of compiling these sort of time series data sets. So this is going to be where I talk about life a little bit. Here we have just some of the more abiotic factors, including temperature here in the red. You can see sort of, understandably, it's warmer in the summertime. You have periods of colder temperatures in the winter. We also have this sort of peak of nutrients in the middle of the summer in this particular system. And then you can see some variability in chlorophyll. We're pretty zoomed out, actually, in terms of the scale bar, but generally overall, we sort of had stable chlorophyll throughout the year. In the spring, there's a little bit more, but nothing, there wasn't akin to huge bloom that happened at any point in the year. But what was really powerful about our approach is that we didn't just stop with, these bulk measurements of chlorophyll. We dug deeper and really wanted to see who is there and potentially, what are they doing. So we used flow cytometry and flow cam analysis, both of which I have here in my lab at UNH. And what we can do from there is really characterize the phytoplankton community composition. And so here I'm showing you how that community breaks out in terms of carbon biomass. So if you can get the size or biovolume of cells, you can convert that into carbon. And so what I want to highlight here is that over the course of our sampling year, we had this dramatic switch between cells that were smaller, so smaller than 20 microns really dominated the system until about the middle of the summer. And then we had a switch to more diatom dominated communities. And here are some images that we took from the flow cytometer, or sorry, from the flow cam. We can also look at depth. So these are grazing rates that we can calculate using the dilution experiment, which is a classic way of capturing more microzooplankton grazing rates. I don't have time to explain how it works, but I'm happy to talk probably ad nauseum about it with you if you would like. And what I want to point out here is that we had variable grazing rates and growth rates between the season, right? So we have time and then the rates here on the y-axis. In general, growth and grazing were higher in the summer versus the winter. And really interestingly, we didn't capture a lot of viral mortality. So there were only 2 occasions throughout the year where we saw mortality events due to viral lysis. And there's a whole host of reasons about that I'm happy to chat about. What we can also get from these dilution experiments are estimations of accumulation rates. And so you can look at, the ratio between growth and grazing and calculate a net accumulation rate. And so when we do that, we saw something very interesting, that we had higher accumulation rates in summer and fall than winter and spring. And what we saw was that the accumulation rate sort of switch happened right around the time where we had this biomass switch. So here's another way of looking at how the biomass changed over time in the estuary. You have your smaller phytoplankton here in blue, and these are just the diatoms plotted here in green. And so if you draw a line down, you have this shift from smaller phytoplankton dominated community to a diatom dominated community right around the time where you had a shift in net accumulation, right? So if you have positive accumulation, that means that your phytoplankton community is increasing over time. If you have a negative accumulation, meaning your population is getting smaller. So the shift to diatoms coincided with a shift in positive accumulation, which meant that phytoplankton were accumulating in the system. And there's a variety of reasons for this. Diatoms can have some grazer avoidance mechanisms. They can produce chemicals or have morphological defenses that can defend them against predation. But we sort of made this linkage between community composition and then rates of consumption. You can then sort of put all this data into an NMDS analysis and look at seasonal shifts in plankton composition. And so this is what we did here, and we sort of color coded them based on time of year. I'm not sure how thrilled my student was with me having him do this, but I think it looks good to me. And what we can do from that is interrogate the data and ask the NMDS, okay, well, what are the species that are responsible for describing these separations of communities over time? And what we got from that really is that Chaetoceros species became very abundant right at that shift in the middle of the summer. And so these cells really described the differences between community structure that we see in the winter, spring, summer, and fall. And likely are very important in driving grazing rates. Okay, so that was a little bit of life and death, sort of using these bulk measurements, but we also wanted to look at life and death using DNA metrics. And so we collected DNA filters. We analyzed those filters for both 16S and 18S amplicon data, so we could get the prokaryotic and eukaryotic community over time. And so what I'm showing you here is the relative abundance of the top prokaryotic communities that we saw over time in our sample. So you have, diatoms and cryptophytes, these really large groups that are commonly seen in the phytoplankton. But what I want to draw your attention to is this group here, the Syndineales. So Syndineales was really the only group that had sort of this structure or seasonal structure throughout the year, right? So we see them increase in relative abundance here in the summer, and they're not as abundant in the colder months. All the other groups have relatively stable relative abundances throughout the year. We don't see really high peaks and valleys. It just so happens, if you don't know, that Syndineales are essentially parasites. They're a large family and group of parasites. They can infect lots of different types of marine plankton, including marine phytoplankton. Dinoflagellates in particular have a long sordid history with Syndineales. And so we sort of followed this rabbit hole down and constructed an entire paper looking at the impact or the interaction between Syndineales and other groups in our ecosystem, so that we're able to try to understand what are the impacts of these parasites on phytoplankton success and phytoplankton abundance and distribution. And I can't go too far into depth with that given time constraints, but the paper is published and I'm happy to talk about it. I will say that Syndinyales, if you are super nerdy and want to read a lot of phytoplankton literature, if you look at any of these sort of big amplicon sequencing or eDNA sequencing data sets, you will find time and time again that Syndinyales is always one of the higher relative abundance groups. So there's something going on there, and there's a yeah.
1 minute, a little bit less
Okay.
than 1 minute. Thank you.
Okay, I'll go quick. So there's something going on there. We can talk about it. Lastly, I just want to leave you with this is a project we're working on now. So we're taking that genetic data and we're looking at how the relative abundance of these communities change over the course of our year. And we're looking at it in terms of temperature. And so we're trying to understand how temperature may drive these communities in the future. And so here's just an example of that. The prokaryotic community, you can see for some groups, their abundance is low in colder temperatures and they increase at higher temperatures. And there are some other really fun shapes that are involved there. There's certainly a seasonal component, but we're trying to use this to sort of help us infer what will happen to these communities as the climate changes. So we can do that for prokaryotes and eukaryotes. These again are, here's your Syndinyales group. So different groups, we can look at that, and that paper is being worked on now. So just to conclude, we're really interested in not just looking at chlorophyll, but looking across groups, really understanding what drives rates of growth and mortality and community functional responses, to really understand how the microbial community will shift in response to a changing climate. And I just want to put this out for general conversation. I'm really excited to think about marine time series in coastal New Hampshire. The really great thing is there's a bunch of them that are currently ongoing, but can we combine forces? And maybe this is already happening. COVID kind of wrecked my entrance into UNH after being here for only 6 months, but if it's happening, I want in, and maybe we can combine forces. And I will put out there that the Harvey lab has been sampling from 2 locations in coastal New Hampshire since October 2020. I have a grad student on that, and we're working up. That data will continue on all of 2021.
Great, thank you, Liz. We have about 1.5 minutes left. That was great.
I'm happy to answer questions offline too.
A lot of great stuff. There's several questions. And I'd like to read from them. And so let's see. So, you mentioned this. How do you anticipate the Gulf of Maine trends in plankton community to be changing as temperature increases and ocean acidification increase?
It's a good question, and I don't think we have I'm going to give a really lame answer. I don't think we really know. I think for sure warmer temperatures are going to benefit some phytoplankton that are happier to hang out in those warmer temperatures, like cyanobacteria, and they will certainly change the phenology of blooms, right? We already see that in the Gulf of Maine. Blooms are happening earlier and at different time points, and that has significant consequences for anything that relies on a bloom.
Great. Do you have any idea about what comprises the less than 20 micron size class that dominated earlier in the year?
Yeah, so that was a combination of picoeukaryotes, nanoeukaryotes, and cyanobacteria. We just lumped them all together. It was in this system, it was primarily picoeukaryotes, so organisms that are between 2 to 5 to 6 microns in size.
There's a couple other questions on here, and maybe we can connect you up with a questioners afterwards.
Sure, for sure.
Possible, Sally? Great. Liz, great presentation.
Thanks, guys.
Thank you very much.
Thank you, Liz. Great presentation. Clearly some overlap in the marine sensing and coastal and estuary resilience research directions. So I don't think you'll have much trouble getting on board with some research directions and research teams and projects. So, and thank you. That was Steve Jones, Associate Director of Sea Grant, taking the time and the questions. And I forgot to introduce myself. I'm Eric Chapman, Associate Director of Outreach for the Marine School and Director of Sea Grant, New Hampshire Sea Grant. Our next speaker is Tracy Mandel. Tracy is an associate professor in mechanical and ocean engineering at the University of New Hampshire. Her research explores several problems in environmental fluid dynamics, fluid mechanics, with a focus on laboratory experiments. She's particularly interested in the surface signature of systems that have complex subsurface hydrodynamics, such as seagrass beds or an oil plume, and how we can develop simplified models that allow us to invert this interior behavior by measuring the free surface. She also studies near-shore circulation and coastal dynamics. Prior to joining the faculty at UNH in January 2020, Tracy received her PhD from Stanford University in the Bob and Norma Street Environmental Fluid Mechanics Laboratory and was a postdoctoral scholar at the University of California in Merced. In this talk, Tracy will briefly describe two projects related to fluid mechanics of flow through vegetation. First, she'll look at the inversion of benthic geometry and flow properties of model seagrass bed from the water surface measurements with implications for mapping and habitat monitoring. And then she'll discuss a new project studying the effects of seasonal plant life cycle changes on coastal protection by salt marshes in New Hampshire. So she's already digging in to local topics. With that, I'll hand it over to Tracy. Thank you.
Hey, thanks for the introduction and thanks for having me today. So yes, as Eric said, I'll be talking about two different projects kind of going from the laboratory flume environment into the field where we're hoping to go in the coming year. And so these two topics, I'll first talk about this question of can the water surface give us information about submerged aquatic ecosystems? And the second topic that I will spend less time on, but hopefully much more time on in the coming year, is how do seasonal changes in salt marsh plants affect their coastal protective benefits? So we'll start with this topic, which is some work from my PhD and some work that I'm hoping to continue here at UNH. And our kind of broader motivation are what are some ways of characterizing the hydrodynamic, that is the way that fluid flows. How does something like seagrass impact the way that fluid flows and interacts with the environment? So one way that we can quantify these sort of effects is looking at, for example, a roughness coefficient. So this is a map of the roughness coefficient of any, exposed, exposed land or wetlands in the lower Mississippi. So this is showing Manning's N, which is a measure of relative bottom roughness or resistance to flow, and is used in a computer simulation. And in this case, these roughness coefficients were assigned using basic land cover definitions. This can be hard to measure in submerged areas and difficult to keep up to date as you have landscape changes over time. We can also characterize roughness by doing detailed benthic surveys, such as the diver that's shown here, but this is cost prohibitive on larger scales, difficult to get, really fine measurements of changes, maybe across from one patch of seagrass to another. And speaking as someone that did scientific dive training in Monterey Bay, where it's 50 degrees year round, 50 Fahrenheit, it's maybe not as appealing to go out and do these sort of surveys in colder water. So the question that I kind of addressed during my PhD was, what if we could tell what is below the water, regardless of water column characteristics, just by measuring the water surface? And so the focus of my PhD work was this sort of bathymetry inversion problem, which asks us, can the water surface give us information about aquatic ecosystems? Whoops, I went the wrong way. Here we go. About bed roughness, or, as someone interested in the interaction of fluid mechanics with ecology, how can it give us information about these sort of aquatic ecosystems like seagrass? And so to narrow this down, I looked at how do interactions between aquatic ecosystems and the surrounding flow show up at the water surface. And so before I could go out into the field and start, taking some measurements of the water surface to try to pick out, there's a seagrass bed here and it's, this much smaller than it was last winter or something like that, how healthy these seagrass beds are. we first need to look at this question in a controlled laboratory setting. And I'm clicking the wrong direction. Our laboratory setup of choice is to use a sort of model seagrass canopy in a flume, which is basically the, I guess, the civil engineer's equivalent of a wind tunnel. We just have some water flowing through a channel. And so I'll show here a video of some dye that's being released kind of in a vertical profile in a current lab student lab setup. So we can see some characteristics of the flow that's showing up. So this is just a way of visualizing what the fluid is doing. You can see we have our model of sort of obstruction, something that's causing flow to slow down, kind of within this model seagrass canopy. And we have much faster moving fluid above that model seagrass canopy. And we can also see a really strong vertical gradient, a strong change in velocity right about the height of these model vegetation elements. So there's an analogy of this sort of flow. In fluid mechanics, we call this a mixing layer, where we have a stream of slow moving fluid and a stream of fast moving fluid that are meeting, and it causes a strong amount of mixing at that point where they're meeting, which in this case is right at the tips of these model vegetation elements. And so it creates these large swirling, overturning fluid structures, basically a vortex. If you hear me say the plural of vortex, it's vortices. So that was what my grandmother asked me at the end of my PhD defense was, what are vortices? So the plural of vortex, just think some swirling mass of fluid, kind of like an eddy. So these swirling masses of fluid are basically shed and carried downstream over a canopy, and they have a very periodic nature. So that's the sort of signal that I decided to look for in the water surface. So to do this, I set up an array of 8 cameras looking down over the length of this model vegetation patch. And what we were able to do was to measure the slope of the water surface over that entire patch over time. And what you will see is something actually very similar to something you might observe out in the real world. So this is a video I took, I think driving home from my grandparents' house or something in upstate New York. And you can see it's a windy day, so we kind of have some constant flow. It's moving more slowly in this field and faster above the field. And you get these ripples, this waving, sort of synchronous waving in this farmer's field that is due to the shedding of these large swirling masses of fluid, these eddies. And you can see there's some amount of, maybe some characteristic wavelength or length scale or periodicity in this sort of flow. So this is a signal that we will be looking for using these cameras. And what we found was actually something that looks maybe very similar to that video I showed. So this is a video on the top. We're looking down at the water surface. So these are changes in the water surface slope. And I have a kind of side view here, just kind of cartoon of what we were observing. And so you can see at the start of this kind of the leading edge where the flow is entering this model seagrass meadow, these vortex structures are still evolving. They're very small. The flow is still adjusting to the presence of some resistance to the flow. And then by the end of that canopy, we can see these bands of light and dark, which are showing positive and negative surface slope. It kind of looks like a wave train or something, but it has very different characteristics from surface waves that you'd see in the ocean. So we see these really, really small amplitude perturbations in the surface slope, and we were able to measure things like the wavelength of this phenomenon, the speed at which this signal at the water surface is moving, the predominant frequency of this sort of vortex shedding. And so we were able to take some surface measurements and connect it to interior flow measurements and properties of our model vegetation. So we ran a number of experiments with, different density of the stems, different height of the vegetation stems, different flow rates and things like that. And what we found, we could compare these. Looks like I failed a little at my animations. So we could look at the flow structure. We'll look at that first. So I showed this kind of sketch of what the velocity, the mean velocity over depth looks like in a seagrass canopy. And we have a sort of within canopy, within seagrass meadow velocity and a velocity near the surface. And so we could measure the speed of these vortex structures moving at the water surface and compare it to a depth average velocity and to the difference between this sort of near surface and within canopy velocity. And we saw that they correlated very well, extremely well for this depth average velocity. There was some more variation due to the relative density of the stems in the canopy, some more scatter due to that. And we also saw that we could connect the vortex size that we measured at the surface, this kind of characteristic wavelength of this phenomenon, to the canopy geometry and to the distance between the tips of our model vegetation and the water surface. So there's kind of some available space for a vortex to occupy. And so that space seems to control the size of this vortex phenomenon we see. And so I won't get into it in this talk, but we were able to then create a predictive model of the mean velocity over depth based just on these surface measurements, so that, in theory, you could go out into the field and say, we see this wavelength of the phenomenon, we see that there are these vortex structures moving at some speed, and then say, these are the sort of velocities that the seagrass is being exposed to, which often controls its, for example, its nutrient uptake rate. Maybe be able to tell that the canopy is extending over some distance, etc. So I'll briefly talk about some of the sort of future directions I'm hoping to take this. And perhaps maybe the most obvious question, maybe a question that all of you have, is how does this surface signature vary with flexible versus rigid vegetation? In my PhD, I used very idealized model vegetation, just some wooden dowels, but you can construct vegetation that will wave that will, I guess, bend within the flow. And so we want to know, is there still as strong a signal? Is there still as strong a predictive relationship between the free surface and interior measurements? We want to know, can we observe this signature in the field? And is it distinguishable from other surface forcing, things like wind and waves? And maybe the sort of big picture here is, can we leverage this to remotely monitor eelgrass habitats in Great Bay? And what sort of remote sensing technology would be appropriate for that? So these are all some sort of ongoing questions. I have a PhD student, Vivek, who actually he started this fall, and he just arrived here at UNH, I guess in the first day of February or something, when we're getting that big snowstorm. So if you eventually see him around Chase, please tell him welcome, and this is the sort of stuff that he will be working on. So if I have time, I have, I guess, just one more slide. 2 more minutes? Okay, thanks. So I'll talk a little bit about another project that's starting up here, looking at the interaction of flow with salt marsh plants and how changes due to the life cycle of these plants might affect whether they are more or less effective at protecting against storms and things like that at different times of year. So this is a project that's that was just funded by Sea Grants Development Funds, which I'm really excited to work on. And so the kind of broad overarching question here is, how do seasonal plant life cycle changes in things like the density of the plants, the stiffness, or how easily the plants break, how do these sort of changes affect overwash and sediment delivery into salt marshes? And this is kind of a project that's, I guess, piggybacking onto some work by Diane Foster and David Burdick and Alison Eberhardt, who approached me when I started and said, is there's some aspect of this work that you think we can, tie into flow through vegetation? So I was really excited to get that opportunity. And so we can kind of see, for example, in these two images from Google Earth of one location south of Rye Harbor, we can see that in April versus October. The vegetation, just looking down, looks very different. It's much more green in fall, probably much more brittle in April, and in some cases kind of covers a different sort of area. And so we want to know for things like on the backside of this dune, if we have more dense, more kind of healthy, vibrant vegetation, will that help reduce the amount that overwash is coming into this back marsh? We also want to know, what are the effects of these plants on sedimentation patterns? If we have some sediment being delivered at different times of year, is it more likely to go further into the marsh at some point because the vegetation has different properties?
Tracy, it's been 15 minutes, so if you can
Okay.
wrap it up.
Okay, I will. This is the last slide. So to do this, we basically want to look at the mechanism that causes vegetation to reduce flow velocities and help sediment settle. And in this case, plant stiffness plays an important role in this process. So we're planning to do some experiments in the mechanical engineering lab to test how flexible or how stiff vegetation is, because that determines basically, does it get streamlined under flow, or does it stay rigid and help dissipate energy? So I will, I guess, close here and happy to take any questions.
Great. Thanks a lot, Tracy. That was great. We do have a couple of questions. One is, do you think the vortices would be so well behaved if the flow is less constrained to be unidirectional and maybe close to constant velocity?
Yes, let me.
This person calls out your picture of submerged seagrass.
Yeah.
Suggesting the flow could spread widely.
Yeah, so I think, there's definitely a question of if you have more realistic sort of flow conditions, if you superimpose waves upon here. I think there have been studies that have seen if you have a combination of waves and currents, you can still see this vortex shedding. But that is a very open question that I'm putting together some proposals on how to answer of, what happens in sort of more realistic flow conditions, and is the signal as strong in those cases?
So can you, pretty straightforward, but probably difficult to answer, how would you use surface characteristics to map presence of eelgrass?
Yeah, that's a great question. And I see the rest of Kala's question there too. So people have no one has done this. But I guess what I would do would look for kind of a predominant frequency that might appear at the surface. There's some predominant wavelength. And there needs to be more work done that would say, how long does it take this signal to develop? And if you have that simplified picture of just a current coming into the seagrass, and then how long does it take that signal to decay once you exit a seagrass bed? So I think those are kind of the main open questions that I believe keep us from being able to, immediately go out into the field and say, there's a seagrass bed and it ends right here. But yeah, if you were able to kind of see this unique peak frequency that we observe at the surface, I think that would kind of help you be able to delineate where seagrass is impacting the flow or where it's at least tall enough to impact the flow and things like that. And yes, it becomes less valuable when the seagrass is deep. There's sort of some limit of submergence where the signal doesn't show up anymore.
Great. Well, I think that's, those are the only two questions. Thank you very much.
Yes, thanks, Tracy. Great talk and excellent questions. You can already get a sense for how those conversations might lead to some pretty interesting places. So as with all the speakers, I encourage you to reach out to these folks and strike up conversations. So these are really great. Also want to point out Diane Foster, the director of the Marine School, has posted a link to the YouTube channel for the Marine School, and encouraging you to follow that station. And that would be great. A lot of great videos there. We'll take you on a tour of many different interesting places that marine research is going on at UNH. Our next speaker is Atsushi Matsuoka. Atsushi arrived in January 2021, not too long ago, and he is a new satellite biological oceanographer research faculty member in the Marine School. He came from Trophic Joint International Laboratory in Quebec City, Canada, where he was the lead of the remote sensing group. He's a biological satellite oceanographer research faculty, and he received a doctorate in the fields of satellite oceanography and marine biooptics from Hokkaido University in Japan. And he's conducted postdoctoral research at a university in Paris, plus at Trophic Joint International Laboratory in Canada. His research activities extend from training intricate in situ relationships between optical properties and microbes, phytoplankton, to monitor much broader scale global climate change from space. His most significant research contributions include establishing fundamental relationships among optical properties and constituents observed in the ocean based on in situ observations, introducing these relationships into radiative transfer model, and developing appropriate algorithms and applying them to satellite data to retrieve and monitor geophysical variables with known uncertainties. In recent years, his research has further expanded to monitor carbon fluxes from permafrost thaw. And with that, I'll hand it over to Atsushi.
Yeah, thank you very much for the nice introduction. As Eric mentioned already, I'm very new, as you can see in my background. Anyway, although my talk is about the Arctic, I'd like to mention that similar approach can be applied to the other coastal regions, such as one in New England. This picture shows, wait a second. So this picture shows the clasping landscape due to the thawing of the permafrost. We can also see the permafrost leaks to the Arctic coastal waters. This is what I will be talking about. This figure shows the trends in mean surface air temperature from 1960 to 2019. What we see here is that high temperature increase, particularly in the Arctic region, much higher increase compared to any other places in the world. This means that global warming is seriously affecting, particularly in the Arctic region. As a consequence, as you know, sea ice of the Arctic Ocean has been decreasing over the last 4 decades. This figure shows the sea ice extent recorded on September 15 last year. For comparison, this yellow line represents median sea ice extent from 1981 to 2010. So as you can see, sea ice has been shrinking over time, particularly in the Arctic Ocean. Recent papers suggested that this dramatic sea ice melt is directly linked to the increase of precipitation. I will come back to this particular topic in the later of this presentation. Over the land, as I mentioned earlier, permafrost is thawing dramatically, as you can see in these pictures. One of the most important reasons why we focus on this topic because permafrost contains a huge amount of organic carbon, which accounts for over 50% of global carbon stocks and more than double that contained in the atmosphere. So there is growing concerns about how much carbon can be released back to the atmosphere as carbon dioxide and methane, which are major greenhouse gases. So permafrost thaw issue is not only for the Arctic, but also for global climate system. So question is, how will organic carbon originating from permafrost thaw influence sea fluxes of CO2 and methane? To answer this question, tracking the organic permafrost originating organic carbon is essential. However, over the land, because of the complexity of the vegetation and soil type, as you can see in these pictures, it might be challenging to track the permafrost originating organic carbon over the land. Instead, tracking organic carbon using remote sensing data in the coastal waters with known uncertainty became recently possible. So this is the main tool that I'm gonna talk about. So we came back to the same question. So as I mentioned, to answer this question, tracking permafrost originating organic carbon, including dissolved and particulate organic carbon from space is essential, which is an objective of this study. So here, data and the method that I used in this study. In situ data includes river discharge, air temperature, precipitation, and active layer depth. Satellite data includes concentration of dissolved organic carbon, DOC, and particulate organic carbon, POC. Here, I note that satellite data are only available from June to September, only 4 months, because of the sea ice cover and the cloud issue. However, data within these 4 months already accounts for over 60% of the total annual flux. So we will be able to keep tracking what's going on using the data within 4 months. Flux is calculated by multiplying concentration by river discharge. It means that flux is controlled by either concentration or river discharge or both. I will come back to this equation along with other figures. So we chose the study area, Mackenzie Delta, north of Canada, as a study area due to the following 3 reasons. First, continuous permafrost is distributed upstream of this region, so we might be able to see the permafrost leaks to the coastal waters. Second reason is that long-term data, meteorological and hydrological data have accumulated in these areas specifically so that we can see what's going on compared to the results obtained in this study. So finally, this area is relatively easy access, so we might be able to examine or confirm our result obtained in this study when necessary. So before I start to show the result, we first compare the satellite-derived DOC flux in red compared to in-situ data in black. I note here that scales in the left-hand side and right-hand side is different. In-situ data shows much higher value. This is because we obtained in-situ data upstream, approximately 200 kilometers away from the river delta. So we expect that less degraded the DOC at the upstream station. The main point from this figure is that satellite DOC flux estimate will capture the natural variability, as you can see here. Now I'm showing you the satellite DOC flux in red and the POC flux in blue. For summer total, it means that I summed up fluxes from June to September for 4 months. When we look at this figure, we don't see any trend over the 20 years, over the last 20 years. However, when we look at the result in September only, when the active layer depth is deepest, we started to see a slight positive trend for both POC and DOC, although these trends are still insignificant in terms of statistics. Here we recall the flux is calculated by multiplying concentration by river discharge. So this is influenced by either concentration or river discharge. Indeed, river discharge rather showed a negative trend for this time period. So it means that concentration should show the positive trend, which is shown in these figures. So now I'm showing you the satellite DOC concentration in red and POC concentration in blue. Previous study using the inch data in the Mackenzie Delta showed that the organic carbon observed in this area, particularly in September, originated from the permafrost thaw using the chemical trace element. So what we see here, the positive trend in both POC and DOC actually suggests that we can see these signatures originating from permafrost thaw from space. POC reflects rather complex processes, including physical weathering, chemical photooxidation, or microbial degradation by marine microbe. So the clear trend is less detectable. However, we observe very significant positive trend even in DOC, which is very remarkable. POC reflects rather physical trend, physical transport of organic matters. So both these results suggest that we can monitor the permafrost originating organic matters in coastal waters from space. To confirm, we also analyzed the long-term meteorological and hydrological data, including river discharge in blue, air temperature in red, thaw depths in brown, and precipitation in gray. We can clearly observe the very strong correlation between air temperature and precipitation, which is very consistent with recent findings. What is the most important feature from this figure is that the thaw depths dramatically increased, specifically since 2011, meaning that hydrological connection is getting more active. To summarize, this figure represents the classic hydrological connection between sea ice, ocean, and atmosphere. So as I mentioned earlier, the hydrological connection is getting more active due to the sea ice decline in recent decades. So reason why, despite the negative trend in river discharge, why we see the positive trend in both DOC and POC, this is most likely due to the major contribution by groundwaters to the coastal waters. And this concentration of organic matter is well mixed in the coastal waters. This is one of the reasons why we see the positive trend of these DOC and POC from space. So finally, I'd like to thank you for the funding resources which contributed to this study. Thank you.
Thank you, Atsushi. Are there any questions? So here's a question. What's the role of coastal surface waves on triggering and the magnitude of permafrost erosion?
I'm not sure if I get your question.
So if you have surface waves,
Oh,
does that,
surface wave.
yeah. Is that, what's the effect on even triggering or the magnitude of permafrost erosion?
This is very important, especially coastal erosion, so coastal permafrost. However, this permafrost in the upstream region, this is insignificant, I would say. And regarding the coastal erosion, contribution might be not that high compared to this upstream permafrost contribution. However, this is going to be more important because of the more coastal erosion is happening in the near future.
Right, great. Can you use your techniques to look at light? And I believe this means light penetration potentially, Kawa?
Yeah.
Yeah. Yes, you can. Okay.
Yes, I can. Yeah.
I think there's going to be some interest in that locally. There's another question. What's the fate of the POC and DOC that does make it into the coastal ocean? In other words, does some of this get released to atmospheric CO2, or is it tied up in the sediments at this low temperatures where it's probably not degraded very rapidly?
Yeah, this is a very good question. So as I mentioned, the DOC has a lot of complex processes involved, and POC as well. And this is the reason why we had a major field campaign in the McIntyre 2019. We use a helicopter to take water from the air, and we also went there using the boat and so on. So there's a lot of processes ongoing, especially for the DOC. For example, like flocculation occur, and photooxidation and microbial degradation. So we are now working on that, how each of these processes contribute to the budget and fates.
Oh, great. This has been a presentation that really lays out some really interesting basic knowledge and great work. Look forward to seeing what you do in the future.
Thank you.
Thanks, Steve, and thanks, Atsushi. A great talk. Clearly, strong relationship folding into ocean sensing and Arctic studies and coastal resilience research directions. And that's it for today. This is normally when we would break and we would go hit the cash bar and our appetizers and mingle. But we're going to take a rain check on that. But we look forward to gathering in person and enjoying each other's company again as soon as possible. I want to encourage people to reach out to the speakers and give them a big New Hampshire Seacoast welcome personally and professionally. I have no doubt that all three of these people will be very popular collaborators as they're able to kind of get closer and closer to resuming our normal life on campus. So thank you very much for coming. We will be having Sally, can you do we have another slide to show our next date or? Nope, that's fine. Our next seminar, we're going to be featuring 2, potentially 3 new speakers on March 16th. Is that correct? Sally?
Yes.
March 16th. All right. So go to our website about the seminar series to learn more, but mark your calendars now and join us for our next seminar later on in exactly a month. Thanks again for coming, and we'll see you in a month.
Thank you all.